It's 10 o'clock, so we might start our seminar. So welcome to the second installment of Polish NMR Seminars. Today we have the very first seminar out of the series of Polish people abroad, and we have Dr. Aneta Lenard from Bayerische NMR Centrum Technical University of Munich, and she will give us a talk called Deciphering the Role of Post-Translational Modification as Key Regulators of Disorder Protein Regions using NMR-based structural biology approach. So Aneta, the floor is yours. I'm stopping the sharing.
Okay, so now I hope that you can see everything.
If you can turn on the presentation mode, it would be great. Yeah, it's
Okay,
good.
then I will decrease you and I will put the laser and I hope you can see and
Yes,
hear
yes,
me.
we can see it.
Great. Then thank you. So thank you very much for inviting me, for kind introduction and giving me opportunity to present my data and take part in this great initiative. So maybe the first question is like how I am related to Polish NMR society. So I'm Polish. I was born and raised in Kraków, where I also obtained my bachelor degree in biochemistry in 2017. Then I moved to Copenhagen in Denmark when I pursue my master's degree, and my master thesis was about investigating the interactions between the extracellular domain of the growth hormone receptor and the growth hormone. And I used different biophysical techniques such as fluorescence spectroscopy, circular dichroism, analytical size exclusion, SACs, and it was the first time I have had any contact with NMR. Then I have moved to Graz, which is the second city, the second largest city in Austria, and it's really beautiful. And I conducted my PhD under supervision of Professor Tobias Madel, and I focused on researching the intrinsically disordered proteins in the regulation, and this will be the topic of today's presentation. Then in May, the May of the last year, I obtained my PhD, and since July, I am in Munich. And I do my postdoc under supervision of Professor Michael Sattler. And now I investigate the big ternary protein complex that is involved in the regulation of alternative splicing. I am still looking at the intrinsically disordered regions, but now I also look at the folded domains that are not so well structurally characterized and their dynamics and the interactions, and also in the context of the full-length proteins. But this is still a bit baby steps, and I can present it in 2 years. So moving on to the topic of today's presentation is I would like to introduce you to famous intrinsically disordered proteins that constitute approximately 30% of a human proteome. These proteins are characterized by the lack of stable secondary tertiary structure and the rapid interconversion between the conformational states. The important thing about intrinsically disordered proteins, IDPs, is that they often serve as interaction hubs, and that is important because those regions are often encountered in DNA and RNA binding proteins, and they serve to regulate processes such as translation, transcription, splicing, apoptosing, and so on. Another important aspect of intrinsically disordered proteins is that they are now more and more recognized to be necessary and sufficient in many cases for a phenomenon called liquid-liquid phase separation, which for people who are not aware of it, it's similar to when you add oil to water and we have a mixture that does not mix together. So this phase separation can be described as the mixing of such homogeneous solution into the two stages, into two phases. One, which is biomolecule enriched phase, dense, and the surrounding biomolecule depleted dilute phase that coexist in the dynamic equilibrium. In cells, the formation of those liquid droplets is characterized by the concentration of of those biomolecules, and it's highly regulated by different environmental stimuli and or factors such as temperature, pH, ionic strength, and more. Why is this important? Is because now those phase separation has been recognized to underlie the formation of membranous organelles, such as stress granule, P-bodies, or nucleoli, but also it is more and more often linked with the formation of a toxic insoluble protein aggregates that then are hallmarks of neurodegenerative diseases such as Alzheimer's disease or dementia or amyotrophic lateral sclerosis, for example. Then I would like to emphasize that in our body, we have approximately 20,000 genes that encode proteins. But the actual number of proteins that we have in our body, it's exceedingly higher because we have splicing, alternative splicing, and also because there is post-translational modifications, PTMs. And those PTMs add another interesting layer of regulation of those liquid droplets. There are many PTMs that are reported in the literature, and we cannot simplify which PTMs act in which way, because the effect of PTM on the phase separation of a given system depends on the PTM and also the system of interest. For example, phosphorylation was reported both to promote formation of the droplets and to suppress it. Another very interesting example is the arginine demethylation that often occurs in the proteins that contains arginine glycine, RGG repeats. And those RGG regions are well known to form the multivalent interaction with the RNA, and that really drives the formation of the liquid droplets. And this can be tuned by arginine methylation. In a couple years ago, it has been proposed that citrination could also play a role. And now I have you. What is citrination? So peptidyl arginine can be converted to peptidyl citrulline when the positively charged guanidinium group is converted to a neutral urido group. And this reaction is catalyzed by protein arginine deaminases, PADS enzymes, that are highly calcium dependent. As a side product, there is release of ammonium. Why those paths are important and this modification. So in our body, we have the family of paths consists of 5 enzymes. And one of PAD6 is catalytically inactive, but those active enzymes, they are expressed in many different tissues, from hair follicle, skin, blood cells to even cells in the neural system. And in the healthy state, the cellular stimuli such as infection or the hormones can activate paths. And this is important because it leads to the proper nerve myelination, immune responses, skin development. But in the state that it's more pathologic and we are in the state of inflammation or hypoxia and the enzymes are too upregulated, it leads to the hypercitrination. And this is often linked with disease states such as cancers or autoimmune diseases such as rheumatoid arthritis, multiple sclerosis, or psoriasis. I have decided to focus on one of the members of the family, PAD4. So PADs can bind from 4 to 6 calcium ions, and the calcium is very important, not only for catalytic activity, but even for the acquiring the proper fold of the protein. And PAD4 is the only PAD that contains nuclear localization signal, which means that the protein can be found both in the cytoplasm, but also in the nucleus. So far, the PAT activity, PAT4, has been studied mostly in the context of citrinating histone proteins or proteins involved in the immune response, such as interleukins. But we thought and we hypothesized that also intrinsically disordered regions and arginine glycine rich region could also be targets for PAT4. And this actually agrees because in the last year, so the project started in 2020, but in the 2014, there's been the paper released, mass spectrometry base, which reported around in one of the mammalian cell line, more than 4,000 proteins to contain at least one PAT4 citrination site, with more than 14,000 sites found. And many of those sites of the for citrination were within intrinsically disordered regions, and the nuclear localization signals of these targets, which came on the good time. So the hypothesis of my project is to investigate not only that PAT4 citrinate, those RGG-rich regions in RNA binding proteins, but also to investigate how it affects different structural, dynamic, functional properties of those substrates, and to look how does it have effect on RNA binding, nucleus, cytoplasmic transport, and arginine methylation. So let's start with the main model system. I focus on the FUS protein, RNA binding protein FUS, which has been reported to form aggregates in amyotrophic lateral sclerosis and in the dementia patients. And I'm interested in the C-terminal region, which contains the RGG3, so the third RGG repeat region, and the P1 nuclear localization sequence. So after purifying proteins in E. coli, the proteins have no modification. But after incubating this in the presence of PAT4 and calcium, well, let's look if the protein is citrinated and how does it look like. So here I will try to have the color scheme when the non-citrinated protein is blue and citrinated is in pink. So when we have a quick look at the one-dimensional proton spectra, we can see most of the peaks or most of the resonances for different backbone protons, aromatics, methyls, they are not affected, but there is an upfield shift for protons delta, and in the citrinated state, as well as for the citrinated protein, we can detect the appearance of the characteristic those peaks, protons eta and epsilon that are here. Then what about the 15N HSQC spectrum, which is fingerprint for proteins? So after the close inspection, we can see that many of the resonance peaks, they are somehow shifted after citrination. There are some differences. But importantly, after citrination, the cross peak corresponding to arginine side chains, well, its signal intensity disappears, but we can detect the two characteristic peaks corresponding to these protons eta and epsilon, which actually, depending on the spectral width and the 15N offset frequency, will be in the different position on the spectrum. And the real 15N chemical shifts for this is 74 and 87 around ppms for protons eta and epsilon respectively. Then when we look at the 13C HSQC spectrum, we can also see that upon citrination, there is the characteristic upfield shift. And what's more, we can also see it from this spectrum, but also it has been assigned. After citrination of this system, I can say that all arginines were converted to citrulline. And in this case, for the citrinated protein, I always have removed the enzyme by the size exclusion chromatography. So we only look at the citrinated state. Then let's look, can we somehow modulate and fine-tune citrination? So then I was interested in looking at the kinetics of this PTM, because NMR provides it's a great technique, well suited to study kinetics of modifications in real time. So only by recording 1D proton spectra over time of the substrate in the presence of enzymes, we can follow how the peaks corresponding to the arginine proton delta start disappearing, and the peak corresponding to the citrulline proton delta start appearing, and the intensity increases. And this can be nicely then plotted against the time. And what I have realized is that after you mix the substrate and the enzyme, the one-to-one, it's super fast. It's like you put the sample and it's already fully citrinated. When you start decreasing the relative enzyme concentration to a bit substoichiometric amounts, like 1 to 4, 1 to 10, it's still very, very fast. So once here we have the 50-fold more of the enzyme of the substrate compared to enzyme, we can see that it takes maybe like within 20 minutes, most of the residues were converted, arginines were converted to citrulline, and maybe around like 5 minutes, like it was like half-half. What about further playing with it? So when I further decrease the enzyme concentration, so the substrate is like 500-fold more, we can see that it takes maybe 1,000 minutes to have most of the arginines converted to citrulline. So it's like overnight experiment. And we can nicely follow this in-between states. And when we plot, maybe after like 80 minutes, we are 50-50. When we, in addition, except playing with the enzyme substrate concentration, we also decrease calcium from 10 millimolar to 1 millimolar. We can see reaction is even more slowed down, and it takes maybe 400 minutes to be like in the between state of the citrinated and non-citrinated. We can also plot the signal intensity. So this is nice, but the other question, which is whether is it only for PAD4? Can only PAD4 citrulinate foods? And the answer is, to answer this, I also express the PAD2 and PAD3, which are the different members of the PADs family, and incubated them with the foods in the presence of calcium. And we can detect that both of those other enzymes also lead to citrination, because we can detect citrulline side chain peaks. But in contrast to the PAD4 mediated citrination, we can also detect some peaks corresponding to the arginine side chains, which can indicate that not all arginines could be converted. So first of all, it could be that there is different substrate specificity, because all those enzymes, they are actually expressed in different tissues, but also it can be linked to a different like catalytic efficiency of those. And PAD4 has been reported to be the most catalytically active and has the highest catalytic efficiency rate compared to other, but it also depends on the specific substrate. So once we know this, another question is that because we use the NMR, and we can also obtain information on the structural and dynamic parameters, the big question is how does citrination even affect structure of fused RGG? So as expected, the RGG region of fused is disordered, and this can be seen because the secondary chemical shifts, they oscillate around 0, and also the heteronuclear NOE, which report on the NH vector bond rotation picot nanosecond timescale. These values are also negative, with maybe like small exception, indicates that the protein is disordered and dynamic, and I don't see any like big differences after citrination. The same when we actually record the nuclear relaxation rates, R1 for a non-citrinated and citrinated state, it's practically the same values. When we plot them against the residue number, and for the R2, I can detect that there is a consistently around 10% lower values for citrullinated proteins, which could indicate some motions at the micromillisecond timescale, but I do not want to speculate more, and we have not investigated further at this point. So the next very important thing is that we wanted to know whether only this is characteristic for FUS, or do other proteins that contains disorder regions and this arginine enriched sequences can be citrullinated. For this reason, we purified many different IDRs from different RNA binding proteins, such as nucleoprotein from SARS-CoV-2, ICP27 from herpes virus, EVS, G3BP1, SAM68, HNRMPA1. And for all of those, we can say that after incubating with PAD4 and calcium, we can detect this, the appearance of the protons eta and epsilon, characteristic for citrullation, and the decrease of the signal for arginine side chains. I cannot say that for all of those we see complete disappearance of the signal for arginine side chains, which can mean that there is still some maybe sequence preferences for certain of sequences or for certain substrates, but at this point we have not investigated this. Our conclusion is that PAD4 can citrullate many intrinsically disordered proteins to different extent. Then another very interesting question is PAD4 can, we can see that PAD4 can citrullate many disordered proteins, but what about folded proteins? Can it citrullate like everything to some extent, at least a little bit? So for this reason, we also took like as example of a small folded proteins that are like not related, not reported to be citrullated, BSA and lysozyme. And after incubating like overnight with PAD4 and calcium, we do not see the appearance of the characteristic peaks for protons eta and epsilon, or the shifts for proton delta, meaning that folded proteins can probably not be citrullated, or not all folded proteins. Then, as an interesting example of the mixture is a forked domain from a FOXO4 protein that contains both alpha helical folded domains and also some disordered tails. And for this protein, we can see that there is still, we can detect some arginine side chain signal in 15-HSQC spectrum, but also we can see some eta and epsilon groups for citrulline, which means there is some of the residues, probably those located in disorder regions, are citrullated, and the rest probably can be not accessible. Then let's move on. What can we learn more? So in our body, as I said in the introduction, we also have other PTMs that are super important for regulation of different processes. For example, arginine methylation. So arginine methylation, it is the addition of or attaching of a methyl groups. It can be either single methyl group attached, or can be two methyl group attached in the asymmetric or symmetric manner. And why is this important? Because, as I said, the arginine methylation often occurs on RGG regions. And we also see that citrullation occurs within those regions. And this is a very important question, whether those two modifications, can they coexist on the same side chain or in the proximity of the sequence or in the space? And do they actually affect each other? So in order to look at this, we have purchased the peptide, synthetic peptide, which contains GRGG repeats, free GRGG repeats, where the arginine in the middle contain one of those methyl groups, so single methylated or demethylated in either way. So to look at this, I have incubated again, for example, the monomethylated GRGG with a PAD4, which you saw in the yellow. So we can see the spectra overlay perfectly. It means without calcium, there is no citrullation. But after adding the calcium, we can see the characteristic upfield shift for protons delta, indicating citrullation. But importantly, what we see is that the signal for methyl groups is not affected, which indicates that the presence of a methyl group is affecting or does not inhibit the citrination of those neighboring arginines, but those methyl groups are itself not like demethylated or converted to some other derivative. So it means that the methylated residues cannot be citrinated. And the same could be shown for those other methyl groups. I have also studied this in the context of the FUS RGG, and we could also see the states in between. So let's move on to the functional implications of citrination. So we know that those RGG regions, they are very important for phase separation to forming those liquid droplets. It often is driven by the multivalent interactions with RNA because those arginines confer the positive charge, whereas the RNA is RNA backbone is negatively charged, so it is very strong, or like yeah, there is multivalent interactions between those, and they drive the formation of those liquid droplets. So when we look at the optical density at like 600 nm, we can observe the changes in turbidity when the protein concentration is kept constant and we increase the RNA. So as expected, for a non-modified proteins, we see the increase of turbidity with higher RNA concentration. But when we perform the same experiment for citrinated variants, there is no change. We don't see any change in the turbidity. And the same can be seen when we look at the microscopy images, because those wild-type, non-citrinated proteins form droplets in the presence of RNA that grow over time, but there is no droplet formation for citrinated. Then the next question is the fact that we can see that citrination affects this droplet formation. Is it linked to RNA binding? Because the phase separation of those RNA binding proteins has been linked to those interactions with RNA. So for this reason, or like we recorded the spectra of the FUS or citrinated FUS in the presence of RNA. As expected, after addition of 0.25 equivalent, molar equivalent of RNA, we can observe the line broadening and also the lack of the signal characteristic for RNA, which can indicate that there is formation of those high molecular weight complexes that have unfavorable relaxation and slower tumbling, and that's why we stop seeing the signal. It can also mean that there is intermediate exchange regime, but probably we can infer that there is RNA binding. When we look at the citrinated form, after addition of RNA, there is no difference. The signal intensity and the position of the peaks, it's the same. And we can still detect the peaks corresponding to RNA, which in this case, I have like 1-to-1 ratio, which is a lot of RNA in the sample, and it is not binding. And the same can be shown for a nucleoprotein and G3BP1. Then the another question is, can we also detect citrination in the context of the full-length protein? This is really cool. So we have the system for the full-length FUS, which contains the different RGG regions and also folded RRM and zinc finger domains, and it has been linked with a solubility tag MBP. This is like 500 amino acids long protein, and it's not easy to study by NMR. When we record the HSQC spectrum, I mean, I think it's predictable that we only see the signals, it will be in blue, corresponding to those disorderly regions, so very narrow proton chemical shifty dispersions, and we don't see any peaks corresponding to the four-folded domains, and it's huge line broadening. It's a very large system. But after addition of PAD4 and calcium, we can see that we see much more cross peaks on 15 HSQC spectrum. Also, the ones ones that can correspond to some folded domains of the protein, and the effect of this line broadening seems to be a bit rescued. So we can say, because we can also see the presence of those cross peaks for the citrulline eta and epsilon side chains, that the protein is citrullinated. Maybe not fully, because we can still see the arginine side chain peaks, but at least to some extent. It could indicate that those are arginines in the disordered RGG regions, at least some of them are citrullinated, and that can disrupt intramolecular interactions, but possibly the arginines, which are in the more folded domains, are not citrullinated. Then the question is, because we know that the full-length protein is citrullinated, and that also the phase separation is suppressed by citrullation, but what about the association with those stress granule, which is one of the membranous organelles? So together with Dorothy Dorman group, they performed for us the stress granule association assay in a permeabilized cells. And we can see, maybe to make it more simple, is that the FUS, so the MVP FUS, is largely localized in the stress granules, and the presence of calcium or PAD4 is not actually changing this. But once the FUS is citrullinated in the presence of PAD4 calcium, its association with stress granule is significantly decreased, which means that the citrullination also affects not only phase separation in vitro, but also recruitment to membranous organelles in cells. Then another big question is that FUS and many other RNA binding proteins, they are important and for shuffling to the new nucleus, so they have to be transported there. And often, for example, FUS is shuffled, is translocated to cell nucleus by interaction with a transport T1, which mediates its nuclear import. So together with our collaborator, they performed the nuclear import assay in also semi-permeabilized cells, after where the cells and the all cell walls were permeabilized, and the nuclear import was blocked with agglutinin, and after adding the EGFP tagged MVP FUS, in the presence of absence of transportin, we can see whether the protein is detected in the nucleus or not. So as expected, without transportin, the protein will not be translocated to the cell nucleus, because it requires interactions with transportin. But when the protein, so the FUS can be detected in nucleus, but once the FUS is citrullinated or in the presence of PAD4 and calcium, its translocation to the nucleus, it's really significantly decreased, which means that the citrullation also affects the transportin 1 mediated nuclear import of FUS. So then we also wanted to look how the citrullation affects the interactions with transportin 1 in vitro. And this is important because transportin 1 is not only crucial for the shuffling of the protein to the nucleus, but the recent studies indicate that it can also act as a chaperone that will decrease its phase separation propensity. So for this, we recorded the spectra of the FUS in the presence of transportin, for the FUS either in the non-modified or in the citrullinated version. So as expected, non-modified FUS in the presence of transportin, we can see significant decrease of many signals, particularly for amino acids in the C-terminal part, which contains this nuclear localization signal, and many arginines within the RGG region. When it comes to citrullinated version of the protein, there is nothing. It's not affected by binding of the transportin. Then the same could have been also seen for G3BP1 and to some extent to nucleoprotein. But I would like to maybe quickly say that this is very interesting example. So the nucleoprotein, so the intrinsically disordered gene-rich region from nucleoprotein from SARS-CoV-2 can interact with transportin 1 because we can see some decrease of intensity of some resonances. When the protein is citrullinated, also we see it's less affected. So there is the intensity of many of the peaks remains unaffected. However, we can actually see that there is some region which could indicate the formation of the transient helix, which is actually not when I designed the construct, it has not been detected. It should not be formation of the helix, but it can be transiently formed. And we can see that there are some of those nests that are not affected. So after addition of transportin, which indicates that there can be some additional binding site, which has been not reported and can involve such interaction via some transiently folded helices, which is independent of a citrulination. So this we can also be predicted with the alpha fold. And this binding, we can also confirm that the proteins in non-citrullinated form are interacting via ITC, so isothermal titration calorimetry, and there is no binding detected for citrullinated proteins. So for this, I can say that with this study, I could show that NMR is very well suited to study protein citrulination and to also indicate how this modification is important for many processes in the cell, such as phase separation, RNA binding, the crosstalk with different modifications or nucleocytoplasmic transport. And very briefly, I would also like to say that I was also involved in the other side project, where we looked at the system which contains like the SIRP, which is called inducible RNA binding protein, and it's arginine RGG-rich region, disordered, which contains RGG, but those RGG are located next to serines. And those serines residues can be phosphorylated in the presence of the cell lysate, but also by a recombinant enzyme, SRPK1. And we can use NMR to study the appearance of phospho residues and disappearance of, in this case, serines over time and obtain information into kinetics of this modification. And as I said, those serines are located next to RGG, RGG regions. So those arginines can be methylated. And this we can also see that they're methylated by PRMT1 enzyme. And also we could study that once the protein is phosphorylated, it affects its methylation because those arginines are located next to two phosphosites, so their methylation is decreased in the presence of phosphorylation of neighboring serines, and we can also see methylation of the residues, which are like far from any possible phosphosite. So we have also assigned side by side. In the other case, when the protein is methylated, so those arginines contains methyl groups, and we try to phosphorylate it, we can see that those phosphosites, which are next to methylation sites, are not phosphorylated. And only one phosphosite, which is far from any attached methyl groups, can be detected. So it is really cool method to study such which site affect what. But it's very important because it's like hundreds of proteins contains RGG regions, and they are often located next to serines. But it is not only phosphorylation and methylation. As I said before, there's also citrullation. So it is very nice tool to study crosstalk between different PTMs. So we have also shown that not only citrullation, methylation, but phosphorylation, those all PTMs can affect the phase separation in vitro, RNA binding, association with membraneless organelles, and interaction with transportin 1. So maybe not linked to NMR, but very briefly in Graz, I had the pleasure to also be involved in the collaboration with Brave Analytics, that is a startup that developed a machine that allows to obtain information of the size and the concentration, like count particles, in the real time online once the sample is pumped, and they measure the optical forces that are trapping the particles in their in the measuring cell. And why is this like interesting is because not only we can when we compare to other size determination methods such as DLS or NTA, this method provides time resolution, and it's also very precise. We can measure each particle, and we can also measure samples which are very polydispersed, such as those phase separating samples, and we can see how it is modulated, for example, this case by addition of RNA. But it's not only for proteins. If you work with some organic samples, organic chemistry samples that are polydispersed and want to know their size, probably you can connect to them. So with this, I would like to thank Tobias Madel Group for their support and all exchanges, our collaborators. Not everyone here is included. And also want to thank my current group. And thank you very much for your attention.
Thank you very much for a great presentation. Erin, we are open for discussion. If you want to ask questions, please turn on your microphone and if possibly camera. If your microphone is not working, you can write the question on the chat. Anyone? Okay, so maybe I'll ask the warm-up question. So for me as a physical chemist, it's always interesting if how much effort it is to prepare the sample itself, not the NMR part, but the preparation, which I think is quite challenging.
Yes, it is the most time-consuming part. It is mostly, protein purification, particularly here. Yes. So it depends on some proteins. Some can be not well behaving and aggregate on the way, and you have to optimize many things. But if maybe it takes, let's say, 1 week, if everything's good, only to purify protein, and then before you need to express it. So like a bit more than 1 week to have like one sample, and then the sample has to be then citrulinated. So the enzyme is not active for a long time. It has to be freshly prepared. And so at first you can prepare your substrates for 1 week in the lab, and then you prepare your enzyme, and then you have to citrinate it overnight, then you have to remove it, and then you run NMR. And especially if you want to have those kinetic experiments, that is a bit painful because you have to optimize a lot of things on the way. But it's thanks to this you start understanding your system.
Any questions from the audience?
So maybe I have a question.
Yes.
So it was interesting what you showed about the study of the full length pus.
Yeah.
Because you showed the HSQC spectra, and the first one showed hardly any peaks. So does it mean that also that C-terminal fragment is folded in the full length protein, or why we don't see all the peaks of the of that unfolded fragment?
Yes. So okay, maybe here because it is much higher signal intensity after citrination, so we cannot see. But basically, it's only like blob, just blob here and blob here and blob here. So it's it would be very difficult and time-consuming another project to only assign to which residues are which. And we cannot just transfer assignments from what we have because it is too much line broadened and too many peaks are missing. So it's very difficult to transfer. Why we cannot see? So we can only speculate, and we think that because I was trying to transfer the assignment, and I think what we see in the full length of the non-modified proteins is mostly some RGG, mostly RGG3, RGG1, and some of the peaks, some serines from the disorder N-terminal part. But it's like some particular residues, and probably they're all involved in the interactions. This is probably a bit phase separating system, so there is intramolecular and intermolecular interactions between different RGGs and with the anti-D and RRM and combinations of those. So only like after citrulinating, so after removing some of those possible interaction sites, then we start seeing those signals. That is actually very like interesting. Like we didn't think. So the same has been in the lab. We have shown for other RNA binding protein, not FUS, but also that we could not see much for the full length. But after citrulinating, this spectral quality improves. So for many of those RNA binding proteins, probably there are those a lot of interactions, and they make it unfavorable relaxation properties, and then citrination can help.
Okay, so also for that unstructured part, you see those interactions when you study full length. Okay, I see. Thank you.
Yeah, but we think that the citrination is probably only in those disordered regions, those RGG. So only those interactions that involve those parts could be affected.
Okay, thanks.
Thank you.
Anyone else interested? Some questions? Let's give it a moment. Okay. It seems that you answered all the questions in your presentation.
Yeah, I think it is. Yeah, I like the project because it is really shows how NMR can be used to study complex biological processes, and we have obtained so many information on like kinetics, site specificity, interactions, crosstalks. Like it is fun for me, but I understand that my talk is very biological. I graduated biochemistry, but NMR is very like it's perfect tool to study this.
Yeah, we have few bio NMR people in the audience, so for sure you reach the good audience here too. Okay, so thank you very much for your presentation, and I'll switch to the PowerPoint. Okay, share. So I would like to invite you all for our next seminar, which will be in 2 weeks. We keep the bio-NMR topic. This time it will be Dr. Vitor Andrawicz from Institute of Bioorganic Chemistry, and he will give us a talk titled NMR Studies of Non-Canonical Nucleic Acid Structures Induced by Specific Binding of Metal Ions. So with that, thank you very much for your attention, and see you in 2 weeks.
Thank you. See you.
Thanks. Bye.